Hello, my dear students. I hope you all have been doing well. Today we are going to discuss the next part of acids, and this is my lecture number 23. And in this lecture, we would be discussing about the titration, pH, and what is the pKa and the Ka of acids, and we would also be discussing about the identification test, the tests that are needed in the laboratory for the identification of acid in the unknown mixture, and we would be discussing about the applications of acid. So the starting from the phenomena of acids means we are going to discuss first about what is the titration. So titration is the procedure. You can say it is a procedure, or you can say it is a phenomena, or you can say it is a method. In that what we do, we add equal number of mole of an acid in a equal number of mole of a base. Means in titration we add equal number of mole of an acid in the equal number of mole of a base. So due to this equal number of an acid and equal number of base, the solution that would obtain by the adding of the equal number of an acid into the equal number of a base will get colorless. So this colorless phenomena is, we say that this is the neutralization. So the titration is the basis of neutralization, and in titration we would be adding equal number of equal number of mole of an acid in equal number of mole of a base. And most probably, most commonly, in acid base titration we would be having um, a we would be having um, a solution of known concentration that we have to add uh, in a solution that is having uh, uh, known volume and unknown concentration. Means what we do in the titration, we will add slowly one solution of a known concentration that we call a titrant to a known volume of another solution of unknown concentration. Means we would for example, if we are adding an acid into a base, so we know about the concentration of an acid. For example, we are adding 1 NaCl, so we know about the concentration of an acid, but we don't know about how much volume of the known concentration of an acid would be required to neutralize the base. So what we will be having, we will be doing in titration, we will be adding a solution that is having known concentration and unknown volume to a solution or a base solution, basic solution, or basic, for example, NaOH is a basic a chemical or basic solution. So we will be adding an acid of known concentration of unknown volume uh, to a base that is having known volume but unknown concentration. So a time reach during this titration when number of mole of an acid becomes equal to the number of mole of the um solution that is having unknown concentration. So what when the number of mole of an acid and number of mole of a base gets equal, what happened? How we know this? This happened to the color change of the solution. So the color change is the indication during the titration. The change of a color of a solution is the indication that in the solution we have added the number equal mole of an acid and in the equal mole of base. So what is the pH? pH is a scale that tell us about how much acidic or how much basic a solution is. means the pH is a indicator, or pH is a scale by which we can identify the acidity or basicity of a solution. So we are having an example here of the pH scale. So you can see this, we are having here different solution and they are having different pH. So how we identify a solution, that is this an acidic solution or a basic solution or a neutral solution? We identify the acidity and the basicity of a solution by this pH scale. So we can see here how it could be happened. Here we are seeing that the this scale is having number from 1 to 4. So all those acidic solution having the pH from 1 to 7. However, all the basic solution having pH from 7 to 14, and the solution that are having the pH 7, these are the neutral solution. So the acidic solution has lower pH and the basic solution have higher pH. You just have to keep in your mind that all those solutions that are acidic in nature will be having low pH. However, all those solutions that are basic will be having higher pH. So we are taking different example. For example, gastric juice, lemon juice, apple juice, tomato juice, all these are acidic solution, and mostly they are sour in nature. And you can see they all are having low pH, and one of the most concentrated or most acidic solution are gastric acid and the lemon juice. And accordingly we are taking different example here. And pure water is having 7 pH, it is neutral, means neither it is acidic nor it is basic. And the from and from 7 to 14 we are having all the basic solution, and these are having higher pH. And bleach is an example of the cleaning agent that we use at our home. So it is strongly basic in nature. And so we see here what is this Ka. Actually, Ka is a term, or Ka is a constant, and we say it as acid dissociation constant. This Ka tell us how strong is our acid. Means in other words, we can say that how strongly a proton have been bind in the solution, means with their other atom in the solution. So if what is uh, pH in other word, we can say this the concentration of proton. So Ka is the means quantity that tell us about how strong is acid, or how strongly proton bind in the solution with other atom. So if a proton is strongly binded or strongly binded with the in the other atom of the solution, it will not be released from and this solution will be less acidic. So we if we are having higher Ka, it means solution is so much acidic. If we are having lower Ka, the solution is not so much acidic. So the Ka is an acid dissociation constant. It tells us how strongly a proton bind with other atom in the solution, and how strong how easily or how strongly proton can be released in the solution. So if the Ka is the strength of a solution, strength of an acid in a solution, Ka tell us how strong is a acid, and Ka tell us the how strong proton bind in the solution, or releasing or not releasing in the solution. If we are having higher Ka, it means the proton are strongly binded with other atom in the solution, it cannot be released. If we are having higher Ka, so it means we are having um, st strong acid. So if we are having lower Ka, it means this acid is not so much strong. If we are having higher Ka, it means uh, the higher Ka means proton are strongly bind with the other atom in the solution and it cannot be dissociate easily. So higher Ka is the strong acid and the lower Ka is the less strong acid. So now what is the pKa? pK is the negative log of Ka. A lower Ka value indicate a strong acid. In the a lower pKa is a strong acid, and the lower pKa is strong acid, and lower Ka is weak acid. Can you understand what I am saying? Lower Ka is weak acid, and lower pKa strong acid. Lower Ka is weak acid means acid is not proton is not dissociating, so we are having less proton in the solution. So lower Ka is the less strong acid and lower pKa is stronger acid. So now we will see this how can we identify acids in laboratory, and here we are taking an example of hydrochloric acid. How we can identify hydrochloric acid in our laboratory? For the identification of hydrochloric acid in laboratory, we use silver nitrate solution. So if we are having an unknown solution, for that we don't know about what is present, what is organic compound present in the solution, and probably we will be having the hydrochloric acid in the solution, but we don't know. So what we have to do, we have to add silver nitrate solution. After the addition of silver nitrate solution, if the silver nitrate solution, if the solution is give us white precipitate, so it is the indication of the presence of hydrochloric acid in the solution. So now we will see that how the acids can be applied in various industries, in food, in pharmaceutical industry, in various other industry. So we will see here one by one. Sulfuric acid is most concentrated example of the acid are used in industries for producing fertilizer, detergent, batteries and dyes, and it also use to remove the impurities from the processing product. And the nitric acid is used to produce salt and fertilizer uh, from the ammonia. When it react ammonia uh, to produce ammonium nitrate, so what we use nitric acid for this purpose. To we use nitric acid to produce ammonium nitrate from the ammonia. So that is the one type of the fertilizer or salt. So in food we use uh, uh, acid as an additives to preserve the taste of the foods. So for example, we use in food sulfuric acid, acetic acid and citric acid. These three acids are more commonly used in food. So sulfuric acid is an example that used in cola. We use this sulfuric acid as preservative in the form of an additive. So and the acetic acid is used in vinegar, and the citric acid is used in pickle. to preserve the pickles. In human body we are having acid in the form of hydrochloric acid, amino acid, fatty acid, nucleic acid, and carbonic acid. So where we found hydrochloric acid in our body, it is present in our GIT tract. So what the hydrochloric acid do is doing in GIT tract? In GIT tract, you know, we are having when we have food, so we have to digest this food. So hydrochloric acid add up there in digestion. It helps to break down complex food into simpler molecule. And amino acid we are having in our body. So amino acid is help us to build a protein in our body. You know, our hair, our skin, and our muscles, all these are made up of protein, and protein made up of amino acids. So amino acids are the acids. Why amino acids are acid? I have previously explained you, because it is having carboxylic acid group. This is an acidic group. So the amino acid in the body help to make the protein from protein, and then protein is used to build up our muscles, to build up our hair, and to produce our nails, like that. And then fatty acid, we also are having fatty acid in our body. We are having fatty acid in our body. Actually, fatty acid are present in the around the cells of our as the cell membrane. You know, there are a lot of fatty acid that are present in our cell membrane of the cell, and they are required for our body for the growth and repair the tissues. And nucleic acid, where nucleic acid present in our body, nucleic acid are present in the nucleus of the cell, and it is important for synthesizing our genetic material. So when our genetic material synthesize and we produce our offspring. So what happened, our cell divide, and this genetic material divide transfer into the next generation, and it is important for build up our traits. So, and the carbonic acid is present in our body fluid, means it is present in our blood, and the carbonic acid maintain the pH, neutral pH of our body, that is 7. What is the pH of our blood? 7. So the carbonic acid is present in our body and it maintain our body pH, blood pH. So now we will see what is the acid catalysis. Acids that may be used as catalyst in industries. For example, in organic chemistry, the sulfuric acid is used in very large quantities for the alkylation, to produce gasoline. Some acids such as sulfuric acid, phosphoric acid, and hydrochloric acid also affect dehydration and condensation reaction. So in biochemistry, many enzymes also use this process of catalysis. So what is this acid catalysis? Sometimes acids are used as catalyst during the chemical reaction. So we say this phenomena as acid catalysis. Acid catalysis. So for this, what acids do, they can do this thing in these can adopt this phenomena in our body. Sulfuric acid, hydrochloric acid, and phosphoric acid can do the catalysis. And now these are some references, and thank you very much for your kind attention.